Our speaker this evening, Dr. Erin Bissell, was part of a military family, and she was raised in several parts of the country, including Montana, Connecticut, and New Jersey. Her bachelor's degree in biology is from Montana State University, and she earned her PhD in the Ecology and Evolutionary Biology Department at CU Boulder, where she studied the genetics of floral shape evolution in wild tobacco. She's been an assistant professor at Metropolitan State University of Denver since 2017, where she has taught courses in general biology, genetics, evolution, plant taxonomy, and field ecology. She's curator of the MSU Denver Herbarium, where she is working to digitize the collection, which primarily consists of plant specimens collected by plant taxonomy students over the past several decades, so the collection can be shared online. In addition to research on bird and vegetative diversity at Chatfield State Park, Dr. Bissell also works with the Denver Botanic Gardens on a riparian vegetation restoration project along Deer Creek. A major focus of all of her research is to provide training opportunities for undergraduate students that will help them find jobs after graduation. Tonight, Dr. Bissell will be reporting on the monitoring of vegetative and avifaunal diversity that she and her undergraduate students have conducted at Chatfield for 4 seasons, both before and after implementation of the Reallocation project. We're pleased to have you, Erin, as our speaker tonight. Erin?
Thank you, Bill, for that introduction. Sorry, things switched over as I started to try to share my screen and I couldn't find the right button for my microphone. COVID problems, virtual problems, I guess is a better way to say it, because COVID problems are far more serious. Thank you, Bill, for that introduction, and the Denver Field Ornithologists for first funding my research and then supporting me by publishing some early results of that research in one of your newsletters, and then inviting me to speak today. Sorry, I'm going to stop speaking for a moment while I remind myself how to do things on Zoom. There we go. I believe that should be sharing now. Is that correct? Okay. One more thing. I'm going to get my laser pointer up. I'm going to go back really quick. I want to give some context for the title I've chosen for my talk, The Birds and the Weeds, Monitoring Ecological Response to the Chatfield Allocation Project. I don't know how many of you, I suspect many of you have spent some time along the Platte River at Chatfield State Park. And I say that because I see a lot of you out birding when we're out doing our data collection for this project. And one thing that I can say about Chatfield is that it's got some really interesting ecotones in terms of, the riparian zone versus the grassland. And then, even though it's not really part of the park, but moving up into the foothills across Wadsworth. But it is rich in weeds. There is no lack of weedy growth at Chatfield, which I think the birds benefit from to a certain degree, but is part of what we're monitoring in this project. So I just want to provide a brief introduction to my talk today. I'm going to start out talking about what the Chatfield Reallocation Project is, for those of you who are not aware of it. And then talk about our sampling design, where the sites are located, what we do in our vegetation surveys, and how that compares to the bird surveys, which are primarily bird counts, and then move on to discussing some of the data that we've collected thus far, as well as the preliminary results that we have from the early analyses. But I also want to take some time to share, what the benefits of undergraduate research are, because programs like the one that DFO, the Denver Field Ornithologists, support are really critical to helping us train undergraduate researchers and graduate researchers for that matter. But at MSU Denver, the focus is on undergraduates. without this kind of money, it's really difficult when, you know, we're spending, most of our time is spent teaching, to obtain the kind of funding that you might get at, say, an R1 research institution like CU Boulder or CSU. And so the funding that's provided from organizations like yours, which are smaller grants, is still critical because it helps support our undergraduates in conducting, you know, these smaller research projects that might not be funded otherwise, and certainly wouldn't gain the attention of, say, NSF, National Science Foundation, or those types of national funding agencies. So I'm really grateful for the commitment that your organization has made to funding those types of projects. And I want to really highlight why I think it's so beneficial, not only to our students, but ultimately to the community, to continue to support that type of funding. All right, so again, you know, this might seem like review to many of you, but to others, it might be completely new. So I'm going to go through it. The Chatfield Reservoir Reallocation Project was a large, rather controversial project that was proposed to increase water retention in Chatfield Reservoir during periods of high water availability. The project itself went through the approval process. The Army Corps of Engineers spearheaded the work alongside partners that included local municipalities, local land management agencies, to essentially alter the amount of water that could be retained in the reservoir. And so what you're seeing in this map is a line that you know, I think pretty obvious. Well, maybe it's not, so I'll just say it. The kind of grayish color here that's shown, that's the current reservoir levels. What is shown in blue are areas that are likely to be inundated under the new water regime. And you know, the reason I was invited to participate in this project was that the Denver Audubon Society approached Dr. Corello, who's actually here with us today, and asked if they knew of anyone who would be able to do a more in-depth monitoring of bird diversity in response to some concerns about the environmental impact statement and environmental approval process that went along with this reallocation project. And in particular, their concern was, you know, for these areas that are shown in part in green, which are cottonwood trees. But I think that the real concern, and it remains my concern as well, although it's been alleviated somewhat, is what's shown in pink here, which are the mature cottonwood trees. These are cottonwood forests that are, you know, anywhere between 50 to the ones shown in pink at least, anywhere between 50 to 90 years of age.
Erin,
Now, certainly there's some, Jim?
Erin, I'm sorry, can you change your view? So to, I believe it's presentation.
Widescreen?
You've got a certain views that we're seeing the next slide. And so the main slide is not as big. It's not taking up the whole screen.
Oh, I did the sharing wrong. I'm going to fix the sharing then. So you're seeing my main screen, not my side screen. That's unfortunate. All right, give me a moment. Thank you for stopping me. I appreciate that. All right, that's what I get. I'm going to confess something. I do this a lot, right? I've been teaching online, but I do it in a different platform. And so it's been a little nerve-wracking to me to think about doing this in Zoom. And then, you know, the people who I teach to, well, I don't know them at the beginning of the semester, I get to know them, so they're familiar to me. And what this forum is lacking is any sort of like facial contact, right? I can't see your response to what I'm doing, but I did just see the thumbs up from Bill. So that's taking up the full screen now? Or was that Jim? I don't know. I don't even know anymore. Yeah. I'm this was exactly what I was worried about, and now I've made the mistake, and so I get to move forward. All right, so going back to what I was saying, you know, the concerns being that these are relatively mature forests that are shown here in pink. The forests that are shown here in green are much younger cottonwood forests that also serve as habitat for birds. But those trees are on average 20 to 50 years old, and likely are new growth since the reservoir was put in, right? So would have grown after the construction associated with the reservoir. Despite the fact that it was approved in 2013, the construction didn't begin until 2017, in part because of planning, but I also think in part because of legal challenges from the Denver Audubon Society. And now, okay, that kind of hung things up for a little bit. And ultimately, the Denver Audubon Society lost those challenges, and construction on the reallocation began in fall 2017. Again, with an eye to trying to clear obstacles that might be associated with temporary higher water line. And I think that that's something that really needs to be emphasized, is this distinction between where the current water line, the current shoreline is found versus where the blue line is found. This is not going to be inundated year round, nor will it be inundated every year. It's going to be variable based on water availability from snowpack and rain. And one of the concerns that, Denver Audubon Society had, and frankly, that I continue to have, is what that type of variation in this zone is going to do to the vegetation and the organisms that are dependent on that vegetation, first and foremost, the birds. And so I started this project in 2015 to get try to obtain some data on both the vegetation as well as the bird diversity and abundance, and I'll come back to what I mean by those in just a moment, in these areas that are going to be in the intermittent flooding zone. So some years they'll, be high and dry, and other years they will be underwater. And we've actually already seen years with them underwater post modification. Again, the reason that this is critical from a Denver field ornithologist standpoint is this is a really critical residential and migratory bird habitat. There are many residential bird species that live either year round or live here in the winter before migrating to their upper summer grounds in the mountains. And then of course, it's an important stopover point for migrants coming from Central and South America up to the northern parts of North America. And because there's water, it's especially critical because it allows them to both find water along what can be a very arid route, but then also habitat, food, shelter, all the things that a bird needs to make a living. In addition to the birds, it's also critical habitat for Preble's jumping mouse, which was taken into account in the plan, the reallocation plan. So they have been negotiating with nearby landowners as well as public land holdings to find suitable alternative habitat for the endangered species, Preble's jumping mouse. Preble's jumping mouse, which is an endangered species. See, this is a point where I would normally look out into the audience and gauge how people are reacting, but I'm just going to keep pushing along. One of the things that I noticed pretty much the first time I visited Chatfield State Park from the mindset of an ecologist is that there are really big differences in stand age between, or differences not just in stand age, but in how the different parts of the forest feel based on stand age. And so one of the things that I did prior to the modification was to look at measures of diversity and community composition that exist between what you see here on the left, what I call stand communities. These are the younger trees that are closer to the shoreline, that typically are somewhere between 20 to 50 years old, and that is based on estimates of the diameter of the tree at breast height. That's what DBH stands for. So, trees add on new growth in rings every year, and their diameter can tell us something about how old they are. What I want to point out to you in this particular picture is just how dense the trees are in this community. This is actually a picture that was taken along Plum Creek, and we've since actually left We don't sample these sites anymore, and I'll explain to you why in just a moment. This was actually after the extensive flooding that occurred in 2015 that left these areas inundated almost all the way up to the road that goes around, that, you know, essentially goes around the park. And so they were underwater for a good portion of the year. And these measurements were taken later in the summer / early fall. Versus what you see in what I call legacy forest, legacy or climax forest. So this is a younger community that you see here on the left, really, really dense trees, really kind of a claustrophobic feeling is how I describe it. Versus if you're on the Platte River, and I want to be clear, there isn't really any legacy forest located along Plum Creek. It's only along the Platte. As you go above Kingfisher Bridge, which is the bridge that separates or that goes over the Platte River, everything to the south of that is legacy forest, and everything to the north of it is, as you move closer to the shoreline, is these young stands that look like this, right? So if I put the bridge here, this would be the south side and this would be the north side. I can show you that on the map here in just a moment. They are what we call a cathedral forest, very open, very airy. They're older, which is why they're so open. So essentially what has happened is succession has eliminated a large portion of these, very narrow trees over time. They die due to disease or wind or snow or lack of water, all the things that trees can die of. And what are left are what we call legacy trees, trees that are very large diameter. I can't put my arms around my legacy trees. I can put my arms around multiple what we call stand trees. And they're older, right? So they're on average 50 to 90 years of age, again, based on DBH, and are characterized by a low stem density. And the reason we call them cathedral forests is because they're open and high, like a cathedral might be, whereas the stand forests are closed and tight and claustrophobic. And that was interesting to me from the perspective of, how is this affecting bird use and bird diversity of these sites? And so just to give you, I don't want to spend too much time here because I'd really like to get to some data, and I'm watching my time. Just to give you a sense of where we started, I told you that we had some stand sites that were on Plum Creek. Those are the sites that were shown up here. But we also had stand sites and continue to have stand sites on Platte River, which again, so this is the southern end of the park. This is the northern end of the park. And right here is Kingfisher Bridge. So we have sites that are located below Kingfisher Bridge to the north that are stand sites, and then legacy, the open sites located above Kingfisher Bridge. We started out with 10 above Kingfisher Bridge, but then dropped that down to 5, and I'll tell you why in just a moment. Likewise, we had Plum Creek for 2016 and 2015, although 2015 data aren't as useful, where the stand sites became inaccessible starting in 2017 due to construction related to the reallocation project. And I ended up choosing to just drop them permanently from the study, just because not being able to access them for 3 years was going to be really problematic from a data analysis standpoint. The modifications, like I said, began in 2017. They didn't start the modifications on the Platte River until 2018. It was really winter 2018, and they were supposed to be done in the spring 2019. But I don't know if you remember what the weather was like in 2019. It was wet, very, very wet, all through March, April, and even into May, which meant that the construction was slowed. And I wasn't able to gain access to these sites again until midsummer. And I think that, the only reason I tell you about that is A, commiserate with me, but more importantly, field work can be, it's at the whim of the elements to a certain degree. And I couldn't safely access those sites until later in the summer in 2019. I've gone through several methods of sampling over the years, based on kind of, what students were doing and then, kind of changing visions for the project. But by 2017, we had settled in on what is called a BACI design, before after control impact. And what that means is that in using a BACI design, you can compare what has happened before and after some sort of modification or impact. And you compare it between sites that are modified, impact sites, versus sites that are not modified, control sites. And so in 2018, well, actually it depends on which site. In 2017, I added control sites at the Denver Botanic Gardens Chatfield Farms, which are, just essentially across a highway. Wadsworth, it's not even really a highway, right? But it's 3 lanes, so it seems that way. Looking, doing some sampling there at 3 different sites. And those are a mix of sites. There's legacy and control stand sites at the Chatfield Farms. But that wasn't enough control sites. And so then in 2018, I added 3 more at Bear Creek, a little further away, which concerned me kind of from a comparative standpoint. But then, I reminded myself, these are birds, they're capable of traversing, distances of that degree pretty easily with relative ease. And the increased sample size, the increased replicants were really necessary for analysis. We do bird counts during the summer months, although because again of problems with access, really we don't tend to start our bird counts. We miss the spring migration nearly every year. And so we've just gotten in the habit of doing our bird counts starting in July, but then running those bird counts through October, so that we essentially have July, August, September, and October, 4 months worth of data. And we sample each site. When I say site, I mean the 5 stands on the Platte River, 5 legacy, 3 control at Chatfield, 3 control at Bear Creek. We sample each of those once a month, so that each site gets sampled 3 to 4 times per season. And then we do our vegetation sampling once per season, typically in the mid to late summer. Once, most of the plants have established themselves. Some of them have already gone to flower and are setting seed, but they haven't necessarily started to senesce. And of course, we miss some of the early spring plants this way, but there's constraints, there's logistical constraints. In terms of this sampling design, essentially we sample at multiple scales. So we sample in our bird counts all birds that are observed within a 50-meter radius of a central point, which is either a stand of trees, if they're young cottonwoods, or one big legacy, if they're the older. We sample every bird that is heard or seen within a 5-minute period of recording. And that's a really strict control, which I think, I know can be limiting in terms of we don't get to record everything that we see, but is important from, again, a replication standpoint, being able to make sense of the data in a meaningful way, year to year, sampling time from sampling time to sampling time. In terms of our vegetation, we also use those very same sites, those very same stands versus trees, to sample all of the trees and shrubs, all of the tree and shrub stems that are within an 11.3 meter radius of the tree. We've since had to modify our sampling design to do an estimate on shrubs, because it turns out that counting every single stem in a thicket of wood rose, or a thicket of, I'm not gonna be able to think of another one that we encounter frequently. Actually, I can. Poison ivy is really, really difficult, especially poison ivy where we don't want to touch it. And poison ivy is rampant in Chatfield. And then within a 5-meter radius, the 11-meter radius is trees, the 5-meter radius is shrubs. And then at the 5-meter cardinal points, we also collect detailed cover data, and when I say detailed, not species, but like percent grass, percent forest, percent shrub, percent tree, that kind of thing. within a one meter diameter circle. So it's kind of these like ever increasing circles to try to get that. And again, the sampling's there so that we can, use the data for analysis. In terms of the vegetation surveys, I just really think it's important to acknowledge that this research is almost entirely driven by my research students. I mean, I offer them guidance, I go out in the field with them, and depending on kind of the level of how long they've worked with me, they have varying degrees of independence. So for the first season, I spent quite a bit of time in the field with them, training them. But then if they have second or third seasons, they not only get to start doing that work on their own, but they also train the new students who are coming in. And I think that's just such a critical aspect of what we do in undergraduate research that, doesn't necessarily, I don't think it's the first thing that springs to people's minds when they think about research. But, these are skills that will serve them when they join the workforce because they're gaining management skills, right? They're getting data management skills and they're gaining people management skills as a consequence of doing this type of work. Kayla and Trevor are two students who were involved with this project right when I got started. They weren't there for the first season. Casey and Ricky were responsible for that season and really got it kicked off. But then Kayla and Trevor stuck around for a couple of years and each of them had different strengths too. And that's another element that I just think is cannot be underemphasized or overemphasized, I guess, is that research, because it's independent, allows students to discover what they enjoy versus what they don't and what their strengths are, right? And really to blossom into, and I mean that as a pun, into, adults who have a better sense of what they want to do based on the experiences they gained through undergraduate research. The protocol I just described to you on the previous page is what's called B-Bird protocol. It was developed by researchers at the University of Montana. It's a pretty standard way of collecting data associated actually with nesting in birds, but it applied here too. And so it was Kayla who actually found that B-Bird protocol and suggested it, and we worked through it together and improved our study design as a consequence of Kayla's research, whereas Trevor really loves data analysis and kind of working out the kinks in the data. So he spent a lot of time on the computer, massaging what the spreadsheets look like so that they can actually, be input into statistical software. The vegetation surveys were funded by a MAR grant from the Colorado Native Plant Society. So again, just really wanting to emphasize the key support that we receive from local organizations. But you're here for the birds, and I know that. In terms of the bird surveys, I've had several students working on those as well. Again, been fortunate to have students work on it for multiple years, although summer 2020 really threw a wrench into the works. I don't know, this summer I feel like I'm starting all over again. And I think Dr. Corello and I are going to be working together. I'll call her Christy because she and I call each other that, working together to try to figure out how to bring students up to speed without the help of other students this time around. The person at the top in this image, Vincent Turco, was the person who was actually supported through the money from Denver Field Ornithologists. I don't know if it's clear to you, but I'm a botanist. I study plants. I'm a plant ecologist and plant evolutionary biologist. I'm not a birder. I'm not a bird researcher. I took a bird identification course in college, and it was not my cup of tea at that moment in time. I didn't say this when I was on the first page, but the reason I always include the picture of the lazuli bunting in my presentations is there was a day when we were out, and I was actually with Vincent, and I believe I was with Gabriel as well, but it might have just been myself and Vincent or Vince. Looking at birds, and we had seen the lazuli bunting, and they have that gorgeous, I'm just going to go back because it's so pretty, why not? They have that gorgeous plumage that's so bright, and to inexperienced birders like me, it's just super pretty, right? This is that. I did not take this photo. really want to make that clear. A woman by the name of Susan submitted it to the Cornell site. But I got really excited. And then we ran into somebody else on the trail who I know is a member of the Colorado Field Ornithologists, and I suspect is also a member of DFO. And she was really excited too. And it was kind of like my first taste of like the thrill that you get when you see a really cool bird. Because I don't remember having that when I was in college, probably because it was early in the morning and I didn't appreciate the opportunity in front of me. I also want to highlight, so the DFO money helped support Vincent, who has more experience, train some of our younger, less experienced students. And they're not always younger. Sometimes they're just less experienced and they're older. So Vincent helped train Gabriel, Amy, and Kayla. And then Kevin Dykstra, who's a former student, also helped, and Rainey as well, helped with training. And in 2017, we had the pleasure of having Joel Such join the group very briefly. Joel is what I call a savant birder. He's been birding since he was 3 maybe. And his auditory skills are out of sight. Like he can hear the slightest variation and, you know, talk. It's amazing. And what working with Joel taught me was that I needed to be helping my students develop their auditory skills better. And so one thing that we've implemented in the intervening years is we actually record the audio for every single count that we do. And, you know, this allows the students to go back and verify their auditory identifications, and they learn from it. So one of my students, Rainey, who's really taken the lead in the last 3 years, realized after listening to the auditory, you know, outside of the field, that she had been misidentifying, I'm not going to get the birds right here. Unfortunately, I didn't put it in my notes, but she had been consistently misidentifying a bird by auditory, because it was just auditory. She wasn't seeing them necessarily. And she was able to go back and correct that data by listening to all of her recordings. And again, that's just, in my opinion, a really critical element of teaching students how to, you know, be better birders, be better bird researchers. I don't want to belabor this information about the data too much, but I think it's important to show some data since that's what you helped me collect. These are data collected prior to the modifications, comparing canopy cover and bare ground between legacy forests and stands. And I just want to point out to you that there's a lot more variation in bare ground cover in the stand than in the legacy forest. And there's a lot more bare ground in the stand than in the legacy forest. So, you know, the canopy is really claustrophobic in the stands, but the undergrowth is not, because the canopy is so closed. And that's what's really being shown here. There's a lot more canopy cover in those stands because there's more stems, there's more trees. They're not as old, they haven't lost as many branches, there's not as many spaces in the canopy to allow light to penetrate to the understory of the forest that allows plants to grow. And that appeared to have some impact on bird abundance. There were more birds, number of birds observed in the legacy forests. In 2016, it was a significant number, a greater number. And then when averaged across both years, it remains significant between the legacy and the stand. So there are fewer birds in the stands than in the legacy. And I'm going to come back to that point in just a moment, because it may be an artifact of how data are collected, but we'll see. And then species richness, which is a measure of the number of species observed. So abundance is the number of individuals. Richness is the number of species observed. Those were not significantly different. So it appears that once the birds make it to Chatfield, you know, they are occurring in both places. And I want to be clear, the birds that we observe are primarily passerines. They're songbirds, perching birds. We don't typically see the raptors that are out along the kind of grassland forest interface because they're not near our sites. Our sites are set back from that. We occasionally get flyovers of some of the shoreline birds like great blue herons. But what we see, the birds we're seeing, are house wrens, mountain chickadees, yellow warblers. They're my favorite because the 4-letter an acronym for recording them is Yahweh, and I'm tickled by that for reasons I don't entirely understand. They're things that you would commonly see in your backyard, frankly, and they tend to be the types of birds that would be, in forest environments. And they don't change. They drop off. The species composition, the number of species drops off as they migrate away. There's actually quite a few resident species, and the resident species tend to dominate year round. Although I haven't done full analysis of that, so I should be careful how I say that. In terms of what has happened after, I'm going to confess I had really hoped to present bird data for after, but without the data from 2020, we just don't have enough for me to do analysis that mean anything. There's not enough power there, is what I might say. There's not enough data there. We'll get more data this year to add to the after to really do the types of comparisons that I would like to do. But right now, we really only have the 2019 data. And the reason I don't have 2020 data is that in the era of COVID, it just wasn't practical to bring students into the field. There were a lot of restrictions on it. And frankly, I know that I'm certainly feeling it today very acutely. I just did not have the bandwidth for it. We've had to transition to teaching our courses online, and it's just been a doozy of a thing, but I'm very excited to get back out into the field this year and to get more data, especially in the after, because we're seeing a different forest in the stands now. I don't know, again, how many of you have been back out, but there was a significant reduction in the number of stems that are present in the stands. You can see the stumps of some trees that were cut. They didn't cut all of them, but the moment I got there, I'm like, this is just different. This is a really radically different place. So the number of trees has significantly declined in the stand forest. In the legacy, there were trees taken there as well, but from a statistical mathematical perspective, the decline was not significant. Perhaps as significant as the number of stems is the canopy cover. Canopy cover has gone way down in the stands. Again, not as significant, and not in fact, it's marginally significant. What we mean by that is there was a decrease in both the stand and the legacy, but the decrease in the canopy forest was more noticeable, and from a statistical perspective, significant. And I'm going to come back to why that matters in just a moment. We also saw a marginally, but not fully significant increase in shrub species in the stands, but not in the legacies. And this is where I was trying to get to. What it comes down to is that tree removal associated with the reallocation plan has reduced canopy cover and likely increased light availability. And that in turn may have released the understory, which includes forbs, broadleaved, herbaceous species that grow and die in a season or maybe last a couple seasons, shrubs and grasses from competition, mostly probably competition for light. And as a consequence, the removal of the trees from the stands may speed up the rate at which they move through succession to a late stage climax force. And I want to remind you that the bird abundance, the number of birds that we observed was greater in the legacy sites than in the stand sites. And so it's possible that this reallocation plan will actually benefit the birds. And again, that's something that I always impress upon my students, that we can't go into our projects expecting to see a certain result or wanting to see a certain result. There's always this temptation that, we're trying to show that, the modifications are going to be bad. Well, bad to who, right? We have to collect the data before we can assess what the effects are. And it may be that we actually see bird habitat improve as a consequence of the changes associated with the reallocation. At the same time, I think it's important to acknowledge some things I'm not measuring. Soil moisture, microbial activity, how is that going to affect the long-term success of these habitats, both of the cottonwoods and of the understory that these birds rely upon? And I don't have the answers to that question, and I'm frankly not collecting the right data to do it. I thought briefly about, should I be trying to collect soil data and then, shipping soil off to the DBG and have them analyze it for fun, and it's like, no, I can't do all the things, right? I have to limit myself to what I can do. Based on what we've seen so far, based on our data from the before modification, bird abundance was greater in legacy sites prior to the modifications. Does that mean that birds prefer more open sites? Maybe because it's easier to find prey and avoid their predators? Or, and this is a problem that could possibly be solved by distance measurements. I'm not sure we have the right data for it, but that's okay. It might be that it's just easier to see birds when the stands are less dense. When there's fewer trees in the way, you can see the birds more easily. And so it might be an artifact of the forest, right? We're just seeing more individuals in the legacy forest than we previously saw. And there's some ways to try to control for that. And I'll try to do it if I can. I'm just not sure I'll be able to. Species richness was not affected by tree density, which seems to suggest that, again, the birds who made it to the habitat used the habitat. They didn't, there weren't differences in which species were where between the two different types of forests. And I hope that remains true post-modification. I do think that there's a place to remain cautious, which is that the increased flooding and frankly, the increased variation in water availability may result in the loss of more trees over time. So right now, what we've seen is essentially, the trees that were part of the controlled culling, right? The people who were in charge of the reallocation modifications marked the least healthy trees for removal. And so in some respects, what has happened is a controlled, controlled logging, I guess might be the word, but that's not what they were doing. Anyway, controlled removal of trees that might have been lost eventually anyway. What remains to be seen is what the long-term effects of variation in water level will look like and how that will impact these sites. And so I have every intention of continuing to collect data until I don't have students to help me collect data anymore. In terms of some student outcomes that I think are really critical, that are really essential, the research experience helps them learn how to develop questions and formulate hypotheses on their own. While, I have my big questions, we constantly talk about, well, what's going on? What are we seeing? What's this new thing? Do we want to add something? Do you want to add something to your part of the project? That kind of thing. And can we actually test that, right? Because it's not enough to ask the question. There has to be a way to test whether that, whatever you're observing is happening, whether it's showing up in the data. And that's where, learning about experimental design comes into play. What does it mean to do field sampling, which is not easy? How do we collect data? How do we manage data? That's a really big question that I'm still, working with. How do we take those data and then, condense them down into 5 slides that I can put into a presentation? Or what you see 2 of my students doing here, Ashley Purcell and Rainey Lamberty, presenting those data at national conferences or at regional conferences or at our school conference, right? We do all levels where they have the opportunity to network, to share their results, and to learn about one of the most important aspects of scientific research, which is communication. This was our Ecological Society of America conference down in New Orleans, which is why everybody's in short sleeves, because it was hot, even in the air conditioning. In terms of their job skills, which I think is another critical element and is, related, but different than just the research skills, because not all of our students are going to go on to research or to graduate school or whatever that looks like for them. I think that they gain critical skills in project design. So learning how to, identify important components for observation, and then how to manage what is happening. How do you prioritize what needs to be done today versus, what can wait until tomorrow? What happens when you're in the field and, you identified your sites in the middle of winter when everything was lacking leaves, and you come back in the spring and find out that everything's covered in poison ivy? Well, you put bags on your legs and figure it out. It also gives them invaluable field experience, which again, whether they use that in their jobs directly or, it just drives a lifelong love and a passion for preservation and conservation, I think both are equally important. But some of these skills are in very high demand in the land management, and in particular by consulting firms who are responsible for determining or helping companies determine where to develop. So several of the students who have worked on this project have gone on to do bird or plant field ID in association with the Bureau of Land Management or with private consulting firms. And then we also do some limited GIS data collection. I haven't yet taken the plunge to do all of our data collection in GIS, but certainly we use it for visualization. And then finally, data management and statistical analysis, which as we continue to collect more and more data on people, whether that's medical data or, ecological data like we're doing here, or data from social media that's used to target people for marketing, having those skills is really important. And whatever they go on to do, they can carry that forward with them. And so I'll just leave with this picture of Ashley and Rainey looking for new sites as we were scouting Bear Creek as a potential study site. I can't remember what we were looking at here, but I will say that Bear Creek is just rich in great horned owls. So it's possible that we were looking at a great horned owl, although the fact that we're along the creek tells me that's probably not the case. They tend to prefer being a little bit further away from the creek. And to really give just a kudos and thanks to my research students who I've tried to mention throughout this, but also people who have helped me with ornithological expertise. Again, not being a bird person, I need it. From former students like Kevin Dykstra, or who at the time was a high school student, Joel Such, Vincent, who was both Vincent and Kevin were graduates of MSU Denver and kind of used this as a springboard into other opportunities. And then of course, my colleague, Dr. Corello, who's been a member of your organization for some time now. And then my funding organizations. And I'd like to point out, they're all small, local, well, I shouldn't say small, there's 150 of you here today. I was a little surprised by that. But local organizations that are committed to supporting research here at the local level. Bill, have you been trying to get me to stop talking and I have been ignoring you? I can't hear you because you're muted, but I will say that I am done now. So.
Oh, okay. All right. Wait a second. Can you hear me now?
Yes, I can.
Oh, okay. We are getting quite a few questions. So
I see that.
For those of you who have submitted questions earlier, I did lose some of them as a result of my technical ineptitude, but we do have some, so I'll start. One of the questions you asked was simply able to see more birds in the less dense stands. Given the possibility of increased visual identification, do you have an idea of what percentage of visual versus oral identification when you were sampling?
So yeah, I mean, definitely, I do think that there's probably a component to the visual, just being able to see more things where there's fewer trees. I haven't run the numbers on auditory versus visual, but I will say that a significant number of our identifications are auditory only or auditory and visual. So again, anything that we hear within that 5-minute window, we either attempt to identify or we count as unidentified. And that's what inspired us, or inspired me, and then told my students to do, to get, actually, it might be that Rainey asked me, I don't recall which, no, I know what it was. Colorado Field Ornithologists annual meeting inspired me to buy a microphone, is what it was, so that I could start to capture that auditory component with a way to go back and check. And so I think that, ultimately I'm going to need to see a statistician to talk about our data. I can do some basic analysis, but that question of, what proportion of the identification is auditory, and are we seeing the same differences with our auditory only identifications, is a really important one. So thank you for the question.
Okay. Another question, would it be interesting or informative to do similar analyses using eBird data? I'm not sure if the caveats associated with eBird checklists would make that data too weird to analyze. In other words, would eBird data be of any help to you in your studies?
Right. So I don't know how many of you speak publicly, but one of the things I always do in preparation for public speaking is think of possible questions. And this was one of them I thought of. I'm like, oh man, eBird data, right? And the reason I thought of it is, I think it's also important to understand, like, we're only capturing a subset of what we see out there within 5 minutes at a particular tree. And we see all sorts of stuff as we're walking around the forest that we record, but that can't be included in our data analysis. With that said, there are many people who are starting to use citizen collected data, whether it's, eBird or iNaturalist. There's all sorts of kind of citizen collected data out there. And there's a lot of people who've been working on how you control for that, what essentially, they're not controlled experiments. So how do you deal with that noise that's in that type of data? And I was feeling really inspired today, probably because I was, it was 8 a.m., 7 a.m., and I was excited about things and I wasn't yet exhausted. That wouldn't it be cool to try to combine some eBird data? Because I know it's abundant at Chatfield with these data that we've collected. And again, I'm going to have to talk to statisticians about that because it's well beyond my skills. And I consider myself decent at statistics, but not that kind of thing. It's hard. You're right. There's a lot of noise in those data. And in part because we can't verify that they're not the same observation over and over again, especially with what birders like to do, right? Which is say, oh, hey, I saw this great, goshawk. I don't know why that came to mind, but it's what came to mind, out at, mile marker 33, you should go check it out. And then everybody puts it into their eBird. Which is that's not a criticism. It's kind of cool, but it's problematic from a data standpoint.
Okay, another question. Will you be analyzing bird population impacts by, looks like goulets? I'm not familiar with that. If thinning selectively removed old stems, it would seem that might reduce habitat for cavity nesters, for example.
I'm sorry, goulage? I was trying to, can you repeat the question for me, please?
GUILDS. Will you be analyzing bird population impacts by GUILDS? I don't know that word either. If thinning selectively removed old stems.
Gills.
Gills? By gills.
Yes.
Gills.
So
okay.
that is on our list of to do things. Analysis by gills, which essentially is kind of functional groups. But again, right now, I haven't had the time to dig into those data. And once again, I'd say that is the type of analysis that I would need to bring a statistician in on.
Okay. Question I have, there's birding records from the banding station just upstream there, south of the Kingfisher Bridge for a number of years. And one of the birds that they recorded there had site fidelity something like 7 or 8 years in a row. What is this? What is your thought of the possibility of what's going to happen to these birds with their habitat being modified as a result of the reallocation?
Because I don't do the banding, I don't actually have any way of determining that. Although, again, gosh, this is just really getting me very excited because I could certainly reach out to the Audubon Society and ask them if we could collaborate on those data at some point. But I don't have the data, the type of data that we collect, I don't have any way of assessing that.
Okay. Could the lack of data from spring migration skew the results of your studies?
So the lack of data for spring migrations, because they're consistently missing, will just mean that my results are only valid for the summer and fall. And the reason the data lack from the spring is because the area is frequently flooded in the spring and we can't access it. So I have data from a couple springs, but the fact that I can't consistently get into the sites in the spring has led me to focus on the late summer and fall. And I mean, it means that I'm not capturing that information, but I am capturing them as they migrate back. And so it's the data I have based on the logistics of what I'm working with.
Okay. Let's see here. This is not a birding question, perhaps not appropriate for your study, but interesting. A couple of years ago, I was on what turned out to be an illegal horse ride. The outfitter didn't know either, on the South Platte, south of the reservoir. I was thrilled to see a pair of river otters. Just wondering if you have encountered them also.
Oh, I've run into people on horses. I have some interesting stories of people on horses. Most of them
otters?
Entirely positive, but certainly some that feel very aggressively about their right to ride. And apparently there are very strict rules around that. So if someone is on horseback, they have right of way. And this was not actually associated with our data collection, it was with the field course that I teach in the same area. We had a tape measure across the trail that the horse did not like, and it led to some elevated voices briefly.
But she's asking whether you've ever seen otters.
Oh, otters.
Otters.
I haven't seen otters specifically on the Platte. I've seen otters on Bear Creek. I have seen beaver dams on Plum Creek. And I have no doubt that there are both beaver and otter along Platte. But in the Platte, one thing that I think is interesting about it, the floodplain is much wider. And so our sites tend to be further away from the river along the Platte than on either, formerly Plum Creek or Bear Creek or Deer Creek.
Where on Bear Creek did you see otters, Erin?
It was on the Chatfield farm site. So they have a resident beaver pair there, but then I saw some otters swimming along the river or along the creek as well, probably in 2018 or 2019, just actually west of Wadsworth. So, kind of the furthest on the Chatfield farm property that you can go. There's a big old tree down there. That's one of our sites that I think we're going to actually lose to the beavers eventually.
Okay.
That's where I saw him.
Okay. Another question. Does Barr Lake, with its widely fluctuating water levels over the course of a year, offer any insights into these questions and concerns about fluctuating water levels at Chatfield?
Yeah, I saw that question and I will confess to not knowing the answer. I haven't read anything about observations made at Barr Lake. And so I don't know if there are actually any data, and that's the beauty of these events, is now I know to look it up. So thank you.
Okay. Well, let's see if we got any more.
Can I address two that I think kind of
Sure.
go with that?
By all means.
So why not use control sites further up the Platte River where the vegetation is undisturbed by the concentration versus using Bear Creek? And then where is Bear Creek location for sampling? So Bear Creek is associated with Bear Creek Lake Park, which is not in the state park system. It's actually through Lakewood. The reason that we use Bear Creek and not the areas that are closer, say down toward Audubon, is there's a change in cottonwood species. So as you move south along the Platte River, you move away from plains cottonwood to lance leaf cottonwood. And to my mind, based on the analyses as we were doing them at the time, I wanted to stick with the same forest species, the same tree species, to try to keep that variable consistent.
Okay. How does looking at other local habitat preserve the Preble's meadow jumping mouse living at Chatfield?
So they've been trying to identify locations. I mean, I have a pretty flippant response to that I'm gonna try to avoid saying out loud. I don't think it does. They try to find locations where habitat is similar, and then argue that habitat is now where the Preble's jumping mouse can live. And the reason I have that kind of flippant response to it is that we visited one of the sites that they had identified as alternative Preble's jumping mouse habitat, that was up. I'm not going to remember the name of the highway. It's not Deer Creek Road. It's the road that's a little further to the south of that. It's not Roxborough. It doesn't matter. My point being, it was an entirely different ecosystem. It wasn't the same at all. It did have a small area where the habitat was wide enough that it might accommodate Preble's jumping mouse, but the likelihood that they would make it there is slim. And if they did make it there, I don't know how well they would do. And so I think that's actually a weakness in our laws that, take into account these and frankly, my data do the same thing. They take into account these kind of gross measures of habitat quality and habitat structure without really thinking about, well, is there any way for the Preble's jumping mouse to actually get here? Because that site was at, I'm not going to remember it was a much higher elevation, and it was definitely a montane, foothills scrub to montane forest habitat instead of the wide floodplain of the Platte River. Nonetheless, there are places along the Platte that they've identified where they're also trying to set aside conservation easements. And the idea is that those would mimic the habitat that you see along the Platte River and offer an alternative site for those endangered species.
You mentioned that in many years, this area will not be flooded. It's only like what it was anticipated, something like 2 out of 5 or something like that.
Yeah, I mean, it's a really small, well, based on historical data, it wouldn't be very frequent, right?
Mhm.
And I don't remember the numbers either. They're pretty small numbers though. It's a small proportion of the time. But I think there's another variable that I don't mention here because frankly, I don't have any data to talk about it. And it's one of the things that I look forward to continuing to observe over the coming decades. And I say that as, committed to this project on that kind of time scale. Climate change can change everything. Climate change changes water dynamics in ways that are really hard for us to predict. And so, if we continue to get these late wet springs, what does that do when these, to these forest communities that require, a certain set of conditions? How does that change the forest composition? And what impact does that have on the animals? And I hope that, beyond the data that are collected for the current question, which again is this reallocation plan, that we can continue, and I'm confident we can, to develop new hypotheses and new questions based on what we're seeing, because it's an interesting community. And it's one of the few, what I would call old growth cottonwood communities remaining. So many of them have been lost to development, which also addresses a question that I'm seeing. And I know you're going to keep telling me questions, but I'm going to answer it really quickly. Cottonwoods live somewhere between, they typically only live to 90, 120 years max. And that's really unusual, 120 years. 90 is kind of like their normal old age.
Okay. Any idea? I know you're a botanist, and it's probably hard for you to answer, maybe, but with the flooding of that area in the legacy cottonwoods, what's your thought of the effect of the reallocation on the legacy trees when they're flooded?
I think it really depends on timing and duration. And so I'm going to start with, I don't know. I'm going to follow that up with, I think it matters when the flooding happens, right? So if the flooding is really early, it might have a different effect than if it's later in the season. And I think how long it remains flooded matters as well. I mean, in the end of the story, cottonwoods are developed. I mean, they evolved to a flooding regime. And what we've done has actually been quite unnatural, which is that we've eliminated that flooding regime, which is necessary for cottonwoods to reseed and produce new propagules, right? They're essentially, their seeds are dispersed and grow, their seedlings grow based on flooding. And so maybe we'll see a kind of a return to something more like normal, or we won't. It won't be like normal, and the flooding will occur at the wrong time. It will last for too long. The roots will be starved, and we'll start to see loss of cottonwoods. I hope it's not the second one. I hope it's the first one, but we'll find
Erin,
out.
Okay, Erin, thank you so much for your very informative talk tonight. I appreciate your coming and explaining what your studies are turning up. And as so often happens with research, you have many more questions as a result of your studies
Yes.
of opening up. So thanks so much for explaining what you're doing. Congratulations to you and your students.
Well, thank you for having me again. I really appreciate it. And if people really feel like they need their question addressed, I'm willing to take those, by email, I guess. I don't know if they probably don't have access to my email, but you can find it on the MSU Denver website. Just be aware that I might not get back to you today or tomorrow. It might be later.
Yes, you can just Google Erin Bissell
Yes.
at Metropolitan State College, and she has her email in that way.